This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 215, recorded on April 9th, 2020. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello, everybody.
How's it going? And you sheltering out there?
Am I sheltered? Man, am I sheltered. I'm sheltered into utter boredom.
I'm sorry. Well, maybe today we can relieve some of that for you.
I am sure you will.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, where we had an incredible hailstorm about an hour ago.
You're kidding?
Really?
No, it was crazy.
It just rained here.
7
We had a thunderstorm.
plagues.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
And here we are all together in a pandemic.
Oof.
A pandemonium.
It's really remarkable. It is really remarkable. All right, so we have a full slate for your listening pleasure today, because probably you would like to listen to some science. So let us start with a snippet, which is going to be all about SARS-CoV-2. I thought it would be interesting to chat for just a bit on an animal model that has been developed to study infection. So SARS-CoV-2, as you know, the virus that's causing COVID-19, the disease, we would like to make antivirals and vaccines, and you really need animal models to test those in. And so mice do not seem to be particularly susceptible to infection. Other avenues are being explored. And the paper I picked to snippetize today is published in Cell, Host, and Microbe. Comes out of Chungbuk National University, University of Southern California, University of Science and Technology in Korea, and St. Jude's Children's Research Hospital in Memphis. And it's called Infection and Rapid Transmission of SARS-CoV-2 in Ferrets. So ferrets are interesting because their respiratory tracts are kind of similar to humans. And what they write here, the anatomic proportions of the ferret upper and lower tract, the density of submucosal glands in the bronchial wall, and the number of generations of terminal bronchioles, that's all the way at the bottom, all reproduce the condition of the human respiratory tract. And in fact, ferrets, did you know that?
No.
I feel like I'm ferreting it out.
We're all ferrets, really. Yes, we are all ferrets.
But
Let me ask you a question about ferrets. Aren't ferrets a favorite animal to study influenza?
They are, yes. They're a good model for flu. They're way better than mice. Ferrets actually will cough. I believe they will sneeze also. And so they're widely studied for influenza.
Question, do they wash their hands?
That is a good question. We'll have to ask them. And so at St. Jude's, they do use ferrets for influenza. And so I think that's part of how this study arose. Now, they took a number of ferrets, and I just want to point out, this is not trivial just to do a ferret experiment, especially not with SARS-CoV-2. So you have to work with the virus in BSL-3 conditions. So you have to have your animals in a BSL-3 laboratory.
Wow.
So you have to apply to do this work. It takes time to get approved. Then you have to breed the ferrets if you don't already have them. So it's not trivial. None of this is trivial. So what you can do,
these were special ferrets that actually had an enhanced human ACE receptor.
That's right.
I'm looking to see what you're talking about here, Michael. I don't think they were special in any way. You're talking about the mice.
The mice are special.
The mice,
that's right.
The ferrets weren't.
No, the mice, in another study, ACE2 transgenic mice, ACE2, of course, is the SARS-CoV-2 receptor. They have been infected, but no clinical symptoms were observed there, and that hasn't even been published yet. So these are just run-of-the-mill ferrets. They take 24 ferrets, and they inoculate them all intranasally. And then they have some that are in the same cage. These are called direct contact or co-housed ferrets. And then they have another set of indirect contact where they put ferrets in cages, separate cages with a barrier, but the barrier is permeable so that air can go through it, and see if you get, transmission by the air.
It's really just a screen, isn't it? Just a screen.
Yeah, it's basically a screen or the cage wall, right? It's a mesh, and they do it 3 times. So 8 ferrets per trial, 3 different times. And here's what they did. They put the virus in their nose, intranasal inoculation. Now, they use what I think is a pretty low amount of virus, 10.5 TCID50. A TCID50 is the amount of virus you need to infect half of a bunch of cell cultures. So I think that's low.
It's 315,000.
How many is that? How many viruses did you say?
Well, it's TCID50. I don't know the relationship between viruses and especially for this particular virus. Michael, you said 315,000?
I think I did the log calculation. It was log 5.5 is what I believe their inoculum was.
So it's not a lot of virus. I mean, and as you'll see, all the ferrets survived, and so I just wonder if they'd used more, it would make a difference. Anyway, they infect the ferrets, and then they monitor them. Their temperature goes up. They go from around 38 to 40 degrees Celsius between 2 and 8 days, and then the temperature goes down. And they note that the ferrets cough occasionally, but they don't lose weight. None of them died. And all of the 6 had increased body temperatures, but nothing past 12 days. So this is a relatively rapid onset of disease. They are able to transmit the virus to each other when they're in the same cage. So contact transmission, they can infect their cage mates. Okay, so close contact. They look at the reproduction of the virus in blood, nasal wash, saliva, urine, feces. The highest amount of RNA, first they do PCR. They find a lot of RNA in nasal wash, so that makes sense. This is how we think the virus is transmitted, from nasal secretion. So there's a lot of virus there. They could also find virus in saliva, PCR products, that is, again. And then they try and isolate virus by putting these washes onto cells in culture. They inoculate the saliva and nasal washes, and they get virus from the nasal wash, but not from saliva specimens. They collected fecal and urine specimens. There's definitely viral RNA in there until 4 days post inoculation.
By the way, can I interrupt for a second?
Yeah,
it's interesting that the in vitro assay on cell cultures, you get plaques with the coronavirus, which is very handy.
Yes, it's a beautiful way to measure real virus, not just pieces of RNA.
Speaking from experience, Vincent?
I am, yeah. I mean, this is an interesting point because, we've transitioned to PCR assays, but it doesn't tell you that there's infectious virus present.
Right.
Yep.
So they can't actually use the urine and fecal specimens to put on cell culture. It kills the cells. So they actually inoculate ferrets with these extracts to see if they can inoculate and get other ferrets infected. So they can do that with nasal wash, and they can do it with urine and fecal specimens. They can infect other ferrets with those. So the point here is that they are shedding virus, and most of it is in the nasal wash, and that can reinfect other ferrets as well. They also looked in some tissues. They find highest virus RNA in nasal turbinates, lung tissue. They could also find RNA in intestine and kidney, which makes sense if they're finding it in feces and urine, of course. But again, nothing is positive after 12 days. And then they look by immunohistochemistry at tissues. They slice up tissues and they put an antibody to a viral protein, and they can see infected cells in the nasal turbinate, the trachea, the lung, intestine of infected ferrets. And they can see immune cells coming into these tissues, especially in the lung. So let's see, is there anything else I wanted to tell you? So they conclude that in these ferrets anyway, the main way that they're transmitted is if they're in the same cage. If they're in separate cages, the other ferrets in the other cage get positive for RNA, but they don't actually shed virus, and so they think that direct contact, at least in their model, is more the way of transmission. Now that's interesting because in people we think it's probably both direct contact and short range nasal droplet shedding
secretions.
But this is an animal model. Even though the virus is replicating in it, it's a model. It's never duplicating everything that happens in people. The real value of this is that you can now use it to test an antiviral. You can get some animal data and go to the FDA and say, look, this antiviral works in a ferret, and now we'd like to do some phase 1 in people. You really need to have that. You cannot just take the activity of an antiviral drug in cell culture and put it in people. You really need animal studies, especially toxicity. You want to know what the maximum dose is for an animal, and then you extrapolate that to people, and then you want to know efficacy in animals as well.
And it's important to point out that in the ferrets, the lung effectively mirrored much of the disease process that we're seeing in people with this SARS-CoV-2 virus.
Yeah, a lot of the kinds of cells infected, the pathology is very similar.
Having said all that, however, recognizing this is a heroic effort, I wonder if this is very practical, because ferrets are much more complicated to grow and to handle in a P3 facility than mice are. And even though the mice that have been reared and which contain the receptor may not be perfect, they're a lot cheaper and simpler.
Yeah, I totally agree. I think it's a very good point. Only certain labs can do ferret studies. That's true. They're expensive, hard to breed, and all that. And so, yes, that will limit it until something better comes out. But right now, we don't have anything better. So
that's right.
So do you have a sense, Vincent, for how many labs worldwide would be able to do these ferret experiments? Like, because of the flu work, is there already an infrastructure in place?
Yeah, there is. There is, I think. In the U.S., there are a number of labs who do influenza in ferrets. In fact, in New York City at Mount Sinai, we have a group that does influenza in ferrets. There's this one at St. Jude's. There are a number of others, and globally as well. So there are labs that are established, and every lab that works on a virus right now seems to be working on SARS-CoV-2. So
right. Well, either
or
that
they're
or
not working at all.
Yes, that's right. I guess most people want to work, so. But I do feel that, and you tell me what you think. I feel that people are really wanting to contribute and do something about this, right? So I think it's an altruistic thing. Anyway, so let's summarize. So the ferret can be infected intranasally. They get high temperature. The virus replicates. They cough, but they don't die. The virus comes in the nasal wash, saliva, urine, and feces. It can be transmitted by direct contact in the same cage, not very well at a distance. And the ferrets get bronchiolitis, which are the bronchioles all the way down into the lung. So I think that's a good model for now to, in certain labs, to study the infection.
That's hopeful. And any hope right now is appreciated.
Yeah, we do need hope, because it's a pretty grim situation out there, right? Virus is spreading unchecked.
Can I say something a little bit on a maybe philosophical side?
You bet.
Okay, well, I had the feeling, what if, imagine that, just for a minute, that the virus came from a different planet. It was imported by evil-minded people who wanted to conquer humanity, and so they brought the virus along. Okay. Now, the characteristic of the virus are that it is really bad stuff, and that our institutions, as we know them, have been at best slow in responding, not because they are evil, not because they are politically inclined that way, but because it's simply a long way towards a vaccine and towards effective preventive or therapeutic drugs. So now, let's say that it goes away. we can expect more. Now we have a mindset that says, yeah, okay, COVID-2 is gone, but something else will come. Okay, so now, and we don't think that our government, our institutions, our scientific institutions, our religious institutions can really help us that much. So this is a different mindset. We're going to be a different kind of human beings, which are going to be fearful, not that somebody's going to invade us from outer space, but that we have the ability, the viruses have the ability to mutate and to do something very bad for us. It's just going to stay with us for a long, long time. That's my point.
Are you trying to set up the next paper from Michele, where we go back in time?
Well,
that's right. That's right.
But I mean, I think it's now incumbent on those of us who are doing biomedical research to become science communicators and educate the public, and also our representatives in government, state and local and federal. I'm more hopeful that we can respond. We do have an election in November, and I think that's going to be critical.
And I have been doing a lot of science communicating and science educating. I have been talking to our local newspaper, both here in Charleston as well as in our capital in Columbia, trying to message the importance of social distancing, because the virus really hasn't adversely affected South Carolina to the same extent that it has knocked out Michigan and New Jersey and New York and California. We have been blessed because of our population density that we haven't yet seen a large number of cases. Now, this is no way to discount it, but we have had a large number of people die in South Carolina. But at the same time, I think it's important for the science community to try to message us so that the general public understands the vital role they play in helping us whip this virus and actually control it.
And we need scientists advising our policymakers and the people who are voting on our budgets.
Besides Tony.
Yeah, Tony can't do it. Yeah. Tony and Bricks, right?
Yeah, the two of them. And she was one of Tony's former students.
Is that right? Well, yeah. I think politics should have no role in any of this, of course, right? You should just listen to the scientists. But unfortunately, as we have seen, it does play a role, and I think that's highly unfortunate. But I would say, Elio, we have had other pandemics in the past, and some of them have been pretty bad. But we didn't learn in the sense that we're not ready. We could have been ready, or more ready than we are. And I don't know why, people think that pandemic preparedness and having vaccines and antivirals ready is not urgent until we're in the situation. I hope that changes, but I am not certain it will. I hope I'm wrong.
Well, I'd like to swing our whole Space Force budget to beefing up our public health infrastructure and our science and healthcare.
Michele, just think of all the trillions they are spending to rescue the economy. If just a fraction of that had been spent on prevention, pandemic prevention, right, and preparedness, we'd be ready. But now we have to pay after the fact, and it's way more expensive. It's highly unfortunate.
And in lives, affecting a lot of people's lives.
If people on the top believe that this was a hoax, what do you expect?
Yes, unbelievable. Yeah.
But I'm also encouraged, Vincent, you and the TWiM team have done a great job of talking about the many avenues of research and how scientists are really coming together, sharing a lot of information to move as quickly as possible to get vaccines and new therapeutics out there. And this paper with the animal model is one example of that.
Yeah, I'm very impressed. It's really great. And I think I saw an article the other day, and they were saying the scientists are becoming the heroes of this story.
Right.
So I would say the scientists and the clinicians, right?
And the people who do the actual caring of patients. Yeah. My God, they're on the forefront of this.
And I'll add that if you would like to learn more about the evolution of this virus, about the immune response to the virus, or about the virus itself and the disease, tune in to This Week in Virology, Immune, and This Week in Evolution, because Vince and his colleagues have been doing a great job providing new material. Boy, it seems like every other day I look and there's more material there. So there's great information out there if you want to learn more.
Yeah.
Thank you. All right. Speaking of learning more, let's learn more from Michele.
All right. I am going to discuss a paper entitled Emergence of Human-Adapted Salmonella enterica is linked to the Neolithization process. This was published in March in Nature, Ecology, and Evolution by a team of 45 authors from 7 countries. The first author is Felix Key. He is a postdoc with Johann Cross and Alexander Hubig, who are at the Max Planck Institute for the Science of Human History in Jena, Germany. And he's also working at MIT in the lab of Tina Lieberman. The other scientists are from 4 different institutions in Russia, 6 or 7 different institutions. 4 institutions in Germany, several in Russia, plus Italy, Switzerland, Turkey, and the UK. And they come from different disciplines: evolution, genetics, anthropology, archaeology, infectious disease, forensics, all to get at the big question: what forces are driving the evolution and emergence of new human pathogens? So, yeah, it's timely given our COVID-19 pandemic.
I was just going to point out that the first author was inspired by a paper that we covered on TWiM in episode 191 that came out describing the Yersinia pestis genome from a 5,000-year-old human remain that was uncovered from an epidemic that was likely in the Bronze Age that left no trace but some genomic pieces in human teeth. And as we'll learn here, as Michele walks us through this incredible paper, we'll see that we can leave traces even from the Neolithic area.
It's amazing. So you could study the evolution of human pathogens in a number of ways. You could study it in a particular person, how that pathogen maybe changes over the course of infection. Or you could study it at the population level. For example, how does SARS-CoV-2 change as it moves across the globe?
But here what they're studying is across human history. And this is history with a capital H. They are using state-of-the-art analysis to look at 8 ancient genomes of Salmonella enterica, which date from 1600 to 6500 years ago. And they compare those genomes to modern Salmonella of today, which you'll have heard about. You probably are familiar with Salmonella typhimurium. It's why we worry about raw chicken or eating uncooked eggs. It can cause gastrointestinal disease, diarrhea, kind of a foodborne pathogen. But what they're able to do is analyze using what they term archaeogenetics to deduce how the human-specific Salmonella paratyphi or typhi, emerged.
So you can follow along. They include a really lovely graphical abstract, which is included as figure 8 of this paper, that really lays out what they're able to learn. So it would have been a heroic effort enough just to study 3,000 modern genomes. And they were able to do that because there's a public resource called the Enterobase, which is a database of more than 300,000 microbial genomes that is maintained by scientists at Warwick Medical School in the UK.
Wow.
But what about these ancient Salmonella genomes? This, for me, was the real wow factor. They were able to obtain remains from human skeletal remains, especially from bones and teeth, and do DNA sequence analysis and comparative analysis.
Carefully.
Very carefully. This is incredibly challenging because, of course, the DNA is scarce. It's damaged. there's a mixed population, and it's likely to be quite a bit different from our modern genomes. So to do this, the team previously had developed a new tool that they call HOPS, which is Heuristic Operations for Pathogen Screening. It's basically a computational biology tool that allows us to use metagenomic analysis, which means genome analysis of a mixture of many different species. And their method is now both specific and sensitive enough that they could accurately identify a species with as few as 50 reads from a sample. So, in other words, this Salmonella enterica DNA was less than 0.001% of the total DNA in the tooth samples that they had. And yet they were able to accurately identify its species and then compare it to our modern day databases. Moreover, they could do this in as little as 1 hour for each run. So this, to me, is really amazing. This method, HOPS, was previously published in Genome Biology in 2019 by the group led by Krauss and Herberg at the Max Planck Institute for the Science of Human History in Germany. So the other question then is, how did they get their samples from? How did they choose the teeth samples to study? Here they relied on studies of ancient civilization that taught us that around the end of the Stone Age, people shifted their lifestyles from a nomadic hunter-gatherer lifestyle to one that was more focused on agriculture and herding grazing animals like sheep and cattle and pigs. So this has been recognized as the period of greatest cultural change in human history about 10,000 years ago. And I'm guessing that was, I don't know what would be number two. Maybe the age of the internet might be the second greatest cultural change. But this is the region that we learned maybe in high school called the Near Eastern Fertile Crescent. And in Figure 1, they have a nice map that shows this is a region that spans from the Mediterranean Sea to the Caspian Sea. And they show on that Figure 1 map where they obtain these samples. The idea already in the literature was that during this period, humans switched from being hunter-gatherers to more herding and having agriculture and having pasture of domesticated animals that they would then use for food. So naturally, then, there would be increased contact between the humans and their the microbiomes of the animals and the feces, and therefore increased exposure to the microbes and swapping both directions between humans and animals. So the thought had been that this period of switching to this more domestic animal and farming lifestyle would have increased transmission of pathogens between humans and animals and given rise to the emergence of human pathogens. So those are the questions they want to get at, but now using more direct DNA-based methods. So their focus is Salmonella enterica, and this is a species that's incredibly diverse. There are more than 2,500 different subspecies or serovars, and typically now we have a lot that are host species specific. So, for example, reptiles have their own variety of Salmonella enterica. Birds have a separate, distinct one. Chickens have a different one. Pigs have a different. And then people, we have a couple different serovars that are now specific to humans. Salmonella enterica typhi or paratyphi. So those are some of the strains that we'll talk about in this paper. What distinguishes the human strains is that they can, rather than just cause gastroenteritis, they can get into our bloodstream and then our lymphatic tissue and spread to deeper tissues and cause quite serious illness. So the question they're getting at here is, when these human-specific Salmonella evolve, and how did they ultimately, they're curious, how were they able to jump from animals to humans and get from the gastrointestinal tract to be equipped to cause more systemic disease? So the current literature is fairly scant, but the leading view is that the Salmonella enterica, a serovar of pigs, jumped to humans as one of the key steps in evolution of the human-specific Serovars.
It's the pig's revenge.
Yeah.
Pig's revenge.
But again, there was not direct evidence for that, but we'll learn how they did it. So their approach was really quite heroic. They had human skeletons that dated from the late Stone Age or Neolithic era all the way to the Middle Ages. They had 2,739 ancient genomes of Salmonella enterica. For 24 of those, they had sufficient coverage. They had 2,700 ancient genomes of from human teeth. And among those, they were able to identify 24 Salmonella enterica genomes. So they then used some enrichment techniques based on DNA hybridization and what's already known about Salmonella enterica, and they were able to amplify and then ultimately assemble 8 complete genomes where they had coverage between 5 and 24-fold of each spot on the genome. So they are can be confident that these accurately represent the salmonella that were circulating these thousands of years ago. So the reason the teeth were so advantageous is that, first of all, they're hard and they're very durable, so they persist over the thousands of years. But also, teeth are nourished by our blood supply, so that any bacterial DNA that they find in the pulp chamber of these fossilized teeth represents bacteria that would have been in the bloodstream, and therefore indicating that the human had sepsis, a blood infection at the time of death, which is what we associate with our Salmonella typhi disease of today.
And also, of course, teeth, the inside of a tooth is well protected, so it's less likely to get contaminated.
Right. So it's a great experimental tool that they have available. So now with these 8 genomes dating from 1,500 to 6,500 years ago, they were able to use the collaboration with their archaeologist and anthropologist experts and align each tooth to the lifestyle of each of the regions where they had obtained these skeletal teeth. So among their 8 genomes, they know that 4 of them came from foragers, individuals who are primarily hunters from 6,500 years ago. 2 of the Salmonella genomes came from people who were living a more domesticated herding animal, herding sheep, herding goats and cattle. So that would have been from the Bronze Age, 5,500 years ago. And then 2 others were more recent from the Bronze Age and Iron Age, and they were from people who did a lot of farming and grazing of animals.
And I take it that you can tell this by looking at the teeth and what part of the tooth is worn by eating, right?
Well, yes, but they also were able to talk with their anthropology and archaeology experts and say, this is where the remains were found. What do you know about? And this is how old they were. What do you know about that period in history? So, in fact, some of the deductions they made from doing the microbial genomics aligned really well with other results that were from studying tools of that era from the Bronze Age and what the distribution of similar tools was across the region. So there was nice congruence between the microbial genetics and the archaeology knowledge that we had from that area.
Isn't that something?
Yeah, amazing. So the first conclusion they were able to draw is that these 8 different genomes were from a widely distributed geographic area, and therefore they could conclude that already by 1,500 to 6,500 years ago, Salmonella was already established. It could get into the human bloodstream. So they want to learn more, and the way they do that was to then compare those 8 genomes to 3,000 additional modern genomes from this Enterobase database. So by doing that, they were, and this is shown in figure 2, they found that really strikingly, the 8 genomes all clustered together on one branch of this huge Salmonella family tree. And they called this branch the ancient Eurasia superbranch. So that was interesting to them. They wanted to kind of test that idea further. So they, first of all, realized that only 2% of all the genomes that are available mapped to this part of the branch. And then they went back and queried the interrobase for additional members of this same phylogenetic branch. And again, they found that the ancient genomes were on part of their own separate, distinct lineage. Therefore, these were likely to be progenitors of the majority of other Salmonella enterica genomes that we have available. And that's shown in figure 2b. So that figure is also really helpful because they illustrate with cartoons. They show the lifestyle of the human host, whether they were a forager or a hunter-gatherer or a farmer. And then they also use cartoons to show what other serovars of Salmonella map to the same branch of the phylogenetic tree or different ones. And they use, for example, little schematics of a sheep, a pig, a horse, even dolphins. So we can look at that figure and appreciate the diversity of Salmonella that have evolved over these thousands of years.
And if I could interject, Felix Key, the first author of the paper, when we queried, perhaps, could you tell us a strong memory of an especially exciting or interesting day in the lab? And as Michele described those cartoons, I'll read you his most memorable day in the lab was perhaps when he obtained these first screening results. All the hidden microbial traces across thousands of samples through 10,000 years of human evolution was fascinating. But as he was printing these large heat maps out across 10 pages and then literally got out the Scotch tape and pasted them together, the molecular evidence for the diseases in the past was right there, front and center of his face. And it was that moment of discovery when you see something that no one else has likely ever seen before. And Salmonella, as we all know, is a substantial pathogen even today in the 21st century. It causes a lot of foodborne illness in the developed as well as developing nations. And so it's been with us for a very long time.
That must have been amazing to peer for the first time way back in evolutionary time to see the progenitors.
Well, then he goes on with his other memory, more specifically about the Salmonella project, is that these phylogenetic results, which often seem very puzzling, because when you look at one of these maps, you sort of scratch your head if you're not deep in the weeds of this technology. But when he started to have a more detailed analysis with the archaeologists, so this is speaking to interdisciplinary interactions, which more and more science is advocating. But talking to the archaeologists, and later they added the ancient human genetics to the samples, the picture became more clear that the different groups we observed in the phylogeny of Salmonella are in agreement with these different prehistoric and cultural groups. And that was the oh wow moment. And it was truly the exciting time to be conducting this science. Normally, when you think about phylogeny, it's you're just looking at, base pair changes. How exciting can that be? But you're really witnessing human history writ large.
And microbial history at that.
That too. The things that can't talk.
And of course, the beauty of this story is it shows how interwoven the evolution of the Salmonella is with the lifestyle history of their hosts. It's very cool. So they were curious then to see if looking at these genomes, they could deduce particular genetic changes that may be accompanied switching from one host to another. So here they were able to rely on a large literature that's been generated by modern Salmonella pathogenesis experiments, where we know that there are a lot of mobile genetic elements that can easily jump from bacterium bacterium and carry particular virulence factors, and then endowing the recipient with new traits that perhaps could allow them to jump and change their residence. So in Figure 3, they have a heat map that shows particular virulence loci and regions that are well studied, and looking at the presence and absence in these ancient genomes compared to more modern genomes. And in general, they find that some lineages are present or absent, some of these virulence factors present or absent, but there are some exceptions. They find, for example, that Salmonella pathogenicity island 6 has both gained and lost loci over time, whereas a different locus called SPV was present in the Salmonella of the farmers and the herders, but not in and I believe it's also now present in Salmonella typhi. So this is a locus that is known to suppress the human innate defenses, and so one possibility is that by acquiring SPV and holding on to it, it would allow the ancient Salmonella enterica to now suppress the innate immune system and allow the bug to survive in the bloodstream and cause deeper tissue infections, which is classic for the human-specific modern Salmonella typhi strain. So that's one approach they're able to use to now better try to understand what genetic events contributed to evolution. The other approach that they use I thought was really clever, and that takes advantage of the known pattern that as a pathogen adapts and specializes in a particular niche, it loses the genes it no longer needs in that new niche. So we call this pseudogene formation, basically acquire mutations in particular loci that you no longer need. And this happens at random, and they accumulate over time. So these pseudogenes act as really nice markers to distinguish one strain from another, basically like scars that accumulate in the genome. So they were able to go through and look at the known number of pseudogenes in all of Salmonella enterica. So for this, they used 8,000 different Salmonella enterica genomes, and they calculated the number of pseudogenes that are present in our modern Salmonella genomes and compared them to the ancient Salmonella genomes. So this is shown in Figure 4a, and it is quite striking because they find all 8 of their ancient Salmonella enterica genomes have the fewest number of pseudogenes. In other words, these ancient genomes have maintained a wider number of genes and therefore more versatility to equip them to live in a range of different hosts. Over time, we see as we look from left to right, we see the accumulation of pseudogenes, and they correspond with host specification. So, for example, then the emergence of the pig-specific serovar correlates with having a large number of pseudogenes. And then the horse-specific serotype has a different set of pseudogenes. So it's a really neat molecular way to gauge the archaeological age and independently calculate the age and deduce the phylogenetic relationship of one genome versus another. So in this case, they found that the number of pseudogenes actually correlated quite nicely with the archaeological age that had been calculated independently from the human remains where the teeth were obtained. So I thought that was really amazing. Among the pseudogenes, they found 2 that were especially interesting, 2 genes that are 2 loci that have accumulated a lot of pseudogenes and independently. These happen to be called FO9 and YDCK. In other words, the ancient generalist Salmonella that could hop among different hosts had functional copies of these genes, but now as Salmonella has evolved and become more specific to pigs or dolphins or humans, they've accumulated independently many mutations in the YDCK and FO9 loci. So this shows us another method that people can use to deduce where a particular genome is on the transition from being a generalist, where it can hop among many different hosts, to being more host specific. And also we can then use it to start to think about the functions that these likely encoded, and maybe begin to understand what the evolutionary pressures were that led to maintenance or loss of these particular loci. It's kind of recreating or deducing the evolutionary pattern. The conclusion then is that this amazing team of 45 different scientists from many countries and many different disciplines were able to deduce that the human-adapted Salmonella enterica paratyphi likely descended from Salmonella that were equipped to jump between grazing animals freely in the Neolithic age, so between farmers and pigs and sheep, etc. And that happened prior to the emergence of the pig-specific Salmonella enterica. So therefore, it means that either humans had the Salmonella typhi progenitor that they then transferred to pigs, where it became pig-specific also, or humans and pigs each independently had acquired a particular lineage that then evolved and accumulated pseudogenes and became adapted to either the pig or the human. So it's allowed them to really revisit what had been thought about emergence of Salmonella. And more generally, I think these methods show us just how powerful that bioinformatics can be, where we can use the DNA of genomes to learn about evolution and emergence of new pathogens. And clearly, there's going to be a lot of interest in this, not only in bacteriology, but in studying pandemics as we're currently experiencing with the COVID-19 illness.
Absolutely. When you consider what's going on, people often compare SARS-CoV-2 to the MERS virus. And the MERS virus has even more frightening case fatality rate of approximately 1 in 3 die from MERS, but unfortunately, or fortunately for us, and unfortunately for the pathogen, MERS doesn't adapt itself well to the human condition. It's much more suited to the camel. And we're trying to understand the significance between MERS and SARS-CoV-2 to really understand if this virus is going to change for the better from the perspective of case fatality rate, or change, hopefully not, for the worse. So this is a very exciting and important field. And I just want to add that Felix has been sort of in the right place at the right time. Like many of us, when he left high school, he didn't really know what he wanted to do necessarily when he grew up. But he writes, being a scientist seemed like a legit profession, and biology always absolutely fascinated him. So he enrolled in a human biology course at the University of Greifswald, a beautiful and fun little town in Germany on the Baltic Sea, where he met his wife and they had their first child. And he's literally led the academic life of moving from lab to lab, and he landed in his PhD lab in the area of archaeogenetics. And one aspect that he particularly loved about archaeogenetics is that it allowed him to ask many interesting questions that absolutely fascinated him. And like I said early in our introduction, he was stimulated at the end of his PhD career by a paper from Simon Rasmussen that came out in Cell in 2015 that we talked about on TWiM 191 about bacteria hiding in the remnants of human teeth. And, from there, he's been moving around the globe. He's been fortunate in that Germany has a very nice program that allows families to do a second postdoc and allowed to move the entire family. And he's lucky in that he has 3 fine children. The youngest was born just recently in January of 2018. And he's actually moved on to his current second postdoc at MIT. And he writes in his advice to young colleagues that what you need to do is be very careful when you pick your mentor, because he writes that the mentor is often someone who can help you really mature, and he cautions us to pick our mentors very carefully because they can actually make the science all that much better and actually help you mature your career like you never thought possible before.
Mm-hmm. Agreed.
That is Felix. And this was a fascinating paper, Michele.
And Felix is moving on to a permanent position, right? He's starting his own group at the Max Planck Institute for Science of Human History.
Yes, he's moving back to Max Planck, and he's going to start there shortly.
That's great.
This idea that the Neolithicism led to emergence, in this case, this is also the same with viruses. The good example is measles virus was thought to have jumped from cows to people about 15,000 years ago when we started to learn how to grow cattle in big numbers and we had contact with them. We got a virus from them, and that's probably the case with many other currently human viruses as well.
Yeah. And even Lyme disease, we shifted to living closer to deers, wanting to, be surrounded by nature. And then we're more susceptible to ticks to jump from the
habitat disruption too. Habitat disruption as we, encroached on their habitat. Their pests actually encroached on our habitat.
Right.
Thank you, Michele. That was great. Lovely.
Thank you, Felix.
Michael, did you want to say one more thing before we wrap up?
I want to say one more thing. COVID-19 claimed one of the giants of microbial physiology and a pioneer in the area of elucidating secondary metabolism last week in Dr. Arnold Domain. Arnie was a force of nature. He published over 500 papers. He was on the faculty of MIT, and he ran fermentation science for many years at Merck. He comes from a family of pickle makers. And if you ever met Dr. Domain, you immediately saw that he had this incredible zest for science and for life. And this was accurately captured in a biography that Dr. Joan Bennett wrote on the occasion of Dr. Domain's 90th birthday in 2017 that celebrated and recounted his accomplishments. And quoting the last bit of Dr. Bennett's article, which was entitled "On Being an Honorary Member of Arnie's Army." And Arnie spelt his name A-R-N-Y, so not to confuse people with Arnold Palmer's Army. Mm-hmm. And some of the musings that Joan wrote in the concluding paragraph with some inspiration from Bob Foljam's list, "In All I Really Need to Know, I Learned in Kindergarten." And here's some of the things we can all learn from Arnie Domain. First and foremost, do good science. Secondly, work hard. Third, publish. Fourth, ask questions. 5, listen. Really listen. 6, do not interrupt. 7, take time to have fun. 8, dance. And Joan, in her biography of Dr. Domain, talks about his love for dance. He loved to dance with his wife. And when he attended scientific meetings, he would often instruct Dr. Bennett to round up some women and they would go out dancing wherever the scientific meeting happened to be. 9, do not take credit for work you did not do, and be generous to your collaborators with respect to the work that they did. 10, do not denigrate other people, even your scientific rivals. Study lots of different things. 12, remember that all microbes are better chemists than we are. 13, also remember Mother Nature had been doing combinatorial chemistry for about 1 billion years. And finally, never, ever forget that natural products evolved for a reason, and that it's our job to find out if their biological activities can be harnessed for human welfare. And that was really Dr. Demain. I got to know him. He was, even at age 81, he served the ASM on a committee that I happen to be a member of. And he was still giving back to our society even then. And I valued his counsel and his suggestions and his wisdom. But what I most remember about him was his joy of life and his love of science. So that was Dr. Demain.
Amen.
Lovely. Thank you, Michael. All right. That is TWiM 215. You can listen on any podcast player. Please subscribe. You get every episode, and we know how many of you are listening. If you like what we do, consider supporting us. Microbe.tv/contribute. Questions and comments, go to twim@microbe.tv. Today we had the whole TWiM team here. Michele Swanson from the University of Michigan. Thanks, Michele.
Thank you, and everybody take care, please.
Yes. Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure. Thank you so much for doing this.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.